The Introduction to Big Data in Life Sciences is a 15-day online program developed by Pine Biotech. In this program, participants will learn about the impact of the genomic revolution, next-generation sequencing, and computational technologies in every area of life sciences, including research, biomedical, biotechnology, and agrobiology. Here, you will learn to find, understand, and then analyze data using high-performance computing, as well as user-friendly tools for biologists. Sessions will be held online and on-site at the Biologic Support Services in Lagos, Nigeria. The program is designed for students and faculty from life sciences backgrounds. In this program, we will speak about big data, how it has transformed the life sciences, as well as introduced both challenges and opportunities into every aspect of research, biomedical discovery, biotechnology, as well as agriculture. The speakers and trainers in this program have ample experience in both academia and industry. Together, they will help illustrate the significance of bioinformatics in every aspect of life science research, as well as show how this discipline can and should become a skill set mastered by every student, researcher, and faculty member. The program will be held online with 1-hour online sessions, as well as self-paced materials that are available for access online throughout our project-based educational portal. The program will start on March 2nd, 2020. In 6 online sessions, we will cover many of the introductory materials that will be important for anyone interested in bioinformatics, data science, experimental design, and research using omics technologies. We will start with an overview of omics data and the significance of the technologies generating it. We will speak about the next-generation sequencing process and how it influences the methods of analysis to process, analyze, as well as interpret the molecular level of information this data yields. The discovery of the structure of DNA had a profound effect on our ability to study the genetic code. However, it took almost 50 years from that time to sequence the first human genome. Soon after, the technology developed at that time evolved rapidly. By 2015, the 1000 Genome Project brought a significant number of whole genome sequences to the research community. And in 2008, over 100,000 whole genomes were sequenced. Today, there is an ever-increasing number of genomic, transcriptomic, proteomic, and metagenomic datasets available that describe subcellular processes with highly detailed digital data. After learning about the history of omics data and its availability, we will speak about the challenges as well as opportunities such data has brought to life sciences research. To illustrate the challenges and opportunities, we will review examples of projects, as well as data types one can find on public domain repositories that are ready for analysis. This introductory course will provide a review of multiple online resources, courses and projects that can serve as a springboard into the world of bioinformatics. The projects include genomic, transcriptomic, metagenomic, and epigenomic data types that you can learn from, and you can even develop a project of your own. In the next session, we will look at some of the information each one of these data types contains, such as single nucleotide variation, gene expression, and even the composition of microbial samples. To understand how these data types are processed, analyzed, as well as visualized, we review the major steps involved in the analysis of NGS data. Omics technologies are becoming cheaper and are used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. Many types of omics data can be generated using NGS, or what is also known as high-throughput sequencing. The data can show detailed information about genomic variants, epigenomic regulation, as well as gene and isoform expression. In the next session, we will spend some time speaking about methods of analysis, as well as computational approaches that can automate the statistical tests we use to find patterns in this oftentimes noisy and complex data. Some of the technologies we will learn to apply are easy, even for the bioinformatics novice, and can be done in Excel or any other type of table manipulation software. Others require a basic understanding of R libraries, as well as how to deploy those in RStudio. We will also discuss more streamlined approaches using the analytical T-Bioinfo platform that we will be using throughout this course. However, the growing amount of variability in the data requires advanced data mining, as well as machine learning that can be sensitive to complexity, and yet extract meaningful patterns in huge datasets. These approaches are regularly used in impactful publications, so we review major types of methods, and then attempt to apply them to several examples from public domain repositories. We will learn about clustering, dimensionality reduction, as well as classification, and the impact of feature selection on interpretation of a condition that is sampled from various datasets. Finally, we will discuss the biology one can find in these datasets, how to leverage reference databases, as well as annotate found patterns by linking objects to genes, pathways, and gene ontology terms, or look for variation between groups and then test for cause and effect relationships in the data. To illustrate these approaches, we will learn to utilize gene set enrichment analysis to find pathways or gene sets that are enriched with up or down-regulated genes of interest. This program will be an excellent introduction to the resources, materials, and a vibrant online community you can join to continue learning about bioinformatics. Specifically, you will get to experience bioinformatics for yourself and try the following. Find and understand NGS data. What is the dataset or project about? What is the question you are trying to ask? And can your data answer the question? You will process data, perform quality control, map and align, then quantify and evaluate pipeline outputs. Next, you will analyze data by learning about the statistical approaches, as well as exploratory data analysis. You will then interpret your data by annotation using reference databases and pathway analysis. Penultimately, you will consider an independent project and have the opportunity to learn more from future OmicsLogic programs online. Lastly, participants will learn about student and faculty resources to make bioinformatics more accessible in Africa. You can review the materials and projects we will use on edu.t-bio.info and request a demo account on the T-Bioinfo Big Data Analytical Platform for bioinformatics analysis. The program is now open for registration and will come with a certificate of participation for those who have completed the requirements for this program. We welcome you to reach out to us with any questions, as well as share this informational video with your colleagues and friends. The program is designed for biologists, clinicians, and researchers, or students that are interested in advancing their understanding of big data and life sciences. The program will not require any background in coding, bioinformatics, or biostatistics. However, we will speak about topics related to cell and molecular biology and utilize terminology coming from biostatistics as well as data science domains. Anyone with a good handle on biological sciences at an undergraduate university level is welcome to join.